10 years ago, to introduce our company to you, I would have told you a story about a cancer patient that was very important to me. I would have told you how cancer had affected their lives and the lives of those around them. Unfortunately, I don't have to do that anymore. Everyone in this room has their own personal cancer story. And if you've paid attention to the news recently, you would have seen there have been some tremendous advancements in the treatment of cancer. But what you haven't seen is an advancement in the treatment of bladder cancer, which is still using 40-year-old technology. Look at a couple of quotes from a recent FDA review panel that had just rejected another bladder cancer drug. Angry, dismal outcomes, unacceptable choices. This is the current state of the treatment of bladder cancer, the fifth most common cancer. Bladder cancer patients deserve better than 40-year-old technology. At Vivimune, our mission is to develop localized cancer treatments that teach the immune system to destroy tumors and prevent their recurrence. My name is Drew McKinnon, and I'm the CEO of Vivimune. I'm going to give you a brief overview of where we're headed today. So it's no secret that pharmaceutical drug development is a risky business. Today we're going to tell you why our solution, our team, and our strategy are set up to mitigate risk, provide returns for our investors, all while getting our solution to the patients that deserve better than those unacceptable choices. This is Sean Smith, our Chief Research and Development Officer, and he's co-inventor of VI-003. He's going to tell you more about our solution and that problem.
So as Drew said, bladder cancer is the fifth most common cancer, but it's still being treated by 40-year-old technology called BCG, which is a bacteria that's placed into the bladder. Unfortunately, many people treated with BCG don't respond, have toxic side effects, or have a bladder cancer recurrence, often leading to the need for full bladder removal. There's a desperate need to develop alternatives to BCG, and now's the time to do it. Here's why. For the last 150 years, we've been limited to three strategies for fighting cancer: surgery to cut it out, radiation to burn it out, or chemotherapy to poison it. Unfortunately, each of these strategies easily miss lingering tumors, and some of them hurt good cells too. Recently, though, a fourth strategy has emerged called immunotherapy, which teaches the body to kill cancer. Immunotherapy is the future, and at Vivimune, we've developed one of these cutting-edge technologies for bladder cancer. Here's how it works. You can think of your immune system like a security force that's constantly screening cells. If it sees a normal cell, then nothing happens. But if it sees a bad mutated cell, then it's quickly recognized and destroyed. Sometimes, though, those mutated cells use disguises to sneak past the immune system. When that happens, they're able to assemble into large cancerous tumors that the immune system can't see. At Vivimune, we develop therapies to reveal those tumor cells to the immune system. Our lead technology, VI-003, consists of an activating protein that raises the threat level wherever it's found, attached to a big sticky polymer that keeps it where it's injected. And this is important because VI-003 is designed to be injected directly into a tumor, raising the threat level only where it matters and prepping the security team to start knocking down doors, ripping off disguises, and destroying the tumor. Once that tumor is destroyed, any disguises that it was using are added by the body into a watch list so that they can't be used anymore to sneak past the immune system. So how does VI-003 compare in bladder cancer versus the standard of care? Well, the standard of care in mice is no better than salt water. But take a look at this early data with VI-003. That top line there, it's not a part of the outline of the chart. It's saying that 100% of mice were able to destroy their bladder tumors and live long term when they were treated with VI-003. Even more exciting is this. Those mice that destroyed their bladder tumors, if we tried to give that same tumor back, even elsewhere in the body, it wouldn't grow, saying that they remembered those disguises that the immune system, that the cancer cells were wearing. So VI-003 has been designed from the start to be a commercializable immunotherapy. Through more than 5 years of development at the National Cancer Institute and in the lab that I work in at the University of Arkansas, Vivimune holds the exclusive option to license the patent underlying this technology, which is currently in the PCT phase of its application. It has 27 claims covering both the product itself as well as its method of production, and we've received a favorable patentability review from a biotech-focused law firm. We have more than 18 years remaining on the life of the patent.
So it's a long way from a patent and mouse data to get into humans. And if I were to summarize the process for you, it'd go something like this. There is a lot of testing, and rightfully so. We've currently been through our preliminary discovery process. We selected our lead molecule, and we've shown that it worked. We're currently doing some preliminary safety testing in small animals, and then we'll move into safety and efficacy testing in large animals before moving to our targeted exit of a phase 1 human clinical trial. So we briefly told you where we are and where we're headed. Now we're going to tell you more about the team that we have in place to help get us there.
I've known everyone on our team for about 5 years, and I got to tell you, Drew McKinnon is a natural at connecting people with solutions. Through his more than 8 years of new business development and medical sales at Cardinal Health and under the umbrella of Intermountain Healthcare, he's been more than prepared to establish relationships with our investors as well as our future acquirers.
Sean Smith, again, is our Chief Research and Development Officer. He's a fifth-year PhD candidate in biomedical engineering at the University of Arkansas. He recently completed an internship at Amgen in their toxicology division, and again, he's co-inventor of VI-003. Sean will be responsible for all things science, and specifically our interaction with our contract research organizations. Corey Costin is our Chief Financial Officer. Corey is a CPA. He currently manages a $9 million budget for the University of Arkansas Honors College. Corey will be responsible for our financials, our accounting, and our compliance.
Dr. Banu Kopulu is currently a postdoctoral fellow in the Laboratory of Vaccine and Immunotherapy Delivery. He is a co-inventor of VI-003, and as our Chief Technology Officer, will continue to develop new intellectual property for the company. Dr. David Zaharoff is our Chief Scientific Officer. He's the lead inventor of VI-003, and he spent more than 5 years at the National Cancer Institute after getting his PhD from Duke. In his current role as an associate professor of biomedical engineering at the University of Arkansas, he's raised more than $3 million in competitive federal grants. He's going to guide our scientific direction. Now, our team has thought carefully about our path to market, and we have identified and addressed 4 main risks. There's product risk in terms of scaling our solution, as well as showing that it's safe and that it works at each stage of testing. There's competition risk in terms of what's already out there, as well as what's in the pipeline. There's regulatory risk in dealing with the FDA, and of course, cash flow. So with these risks in mind, we've framed our strategy around 4 main decisions, starting with our decision to target bladder cancer. So we do have great data in bladder cancer, as you saw, but we also have promising data against other tumors, such as pancreatic, colon, and breast tumors. But we chose to start with bladder cancer and then move into those other indications for 3 reasons. First, as we already mentioned, bladder cancer is currently being treated by an old, low-efficacy technology, setting the bar low for regulatory approval. Second, targeting bladder cancer allows us to mitigate competition risk by entering into a relatively thin competitive landscape. So normally you want to be one of the big bars in one of these charts. But in our instance, it's to our advantage to target the relatively thin bladder cancer immunotherapy landscape with only 13 new drugs in development. And of those, if you compare them to VI-003, we are the only one that not only puts it locally, but keeps it there. And what this means for patients is fewer injections, fewer doctor's visits, and reduced toxicity. And what it means for our eventual acquirer is the need for supplying less material per treatment. Even a bigger deal is that VI-003 has the best preclinical data demonstrating the body-wide protection that we talked about earlier in terms of remembering those disguises in a way that's unique to each patient. So we have an old technology and a relatively thin competitive landscape, paving the way for an easier entry into the bladder cancer market that we estimate to be around $11 billion. And once again, this is just the start. An approval in bladder cancer can then ease the transition of VI-003 into other indications such as pancreatic, colon, and breast cancers, leading to the potential for huge market growth.
So to mitigate product and regulatory risk, we'll continue to do as much of the preclinical development and discovery work in our own in-house lab, and then we'll bring in outside expertise when necessary, specifically a contract research organization to help us with our toxicology studies, and a contract manufacturing organization to help us scale our product. Now, we've talked with multiple CROs, and we've selected Charles River based on their expertise and their success with getting oncology drugs through the FDA. We've also selected Cytovance Biologics to help us scale our product, again based on their expertise and their knowledge of the different components of our immunotherapy.
So the third part of our strategy is to leverage our scientific network and our advisory board. So as I mentioned, Dr. David Zaharoff, our CSO, spent 5 years at the National Cancer Institute. We can use his relationships there to establish a collaborative research and development agreement, or CRADA. What this does for us is it allows us access to the NCI's network of physicians and patients, builds credibility around our technology, And as you can see, it significantly reduces the phase 1 costs. We also have an experienced advisory board with expertise in biotech development, in dealing with the FDA, as well as in venture capital. But I'd like to highlight Dr. Neal Shore. He's a world-renowned urologist with over 100 clinical trials to his name, and he is a substantial asset to us in terms of building credibility around our technology.
So the fourth part of our risk mitigating strategy is to sublicense our technology early to a pharmaceutical company. When you think about any exit, there are a lot of things that go into determining when and if that exit happens. And in our case, we have some built-in advantages based on the market that we're in. The first advantage is that pharmaceutical companies consistently have to deal with the problem of their patents expiring. This has led to the common industry practice of acquiring small biotechs like ourselves in order to grow the size of their pipeline. The second advantage is just the tremendous growth of the immunotherapy market in general, as evidenced by the $10 billion in deals that happened in the space in last year alone. So again, our decision to sublicense our technology early is strategic. We will have assumed the risk of getting our therapy through to the clinic, but in doing so, we will have substantially increased our value, which we estimate to be $500 million after successful phase 1. So when you consider the current immunotherapy landscape, that $500 million valuation is reasonable. As with any deal, there are a lot of things that go into the determination of value, and in our case, we consider the tremendous growth of the immunotherapy market, as well as the comparable deals that you see listed here, which are all platform immunotherapies that have multiple cancer indications. As you can also see, we've received feedback from Big Pharma indicating that not only is our technology unique, but also that our decision to sublicense our technology early is the right one. So how do we get to that valuation? Well, in addition to the $4 million and 5 years of research that's already gone into the development of the product at the National Cancer Institute, big picture, we need $9 million in approximately 5 years to get us to that phase 1. So there are multiple exit points for us along the way. And with the $2 million in angel investment that we're seeking, coupled with the $2.3 million in SBIR grants that we will apply for, we can get through 2 of those major milestones, which are proving we can scale the product and proving that it's safe in animals. So not only does this provide us an exit point after only 2.5 years, but it also positions us to go after our Series A round of $4.7 million of institutional capital. With that Series A, we can get through full-scale production and through our phase 1 clinical trial, which gets us to that final valuation, and that's a 45x return for our angel investors. So a 45x return is exciting for angel investors, but there are other things that are going on in the market that are equally as exciting. You may have seen that President Jimmy Carter was recently declared cancer-free after using a combination of an immunotherapy and radiation to treat his cancer. We briefly touched on it before, but the deals that have happened in this space were north of $10 billion last year, and the momentum has continued to build based on the innovation that's taking place in the space. And finally, during the President's State of the Union address, he announced the development of the National Cancer Moonshot, which opens up data and information sharing across clinicians. It provides an influx of capital into grant funding for cancer research, and specifically immunotherapies. And it also targets easing the regulatory burden of the FDA, which helps companies like ourselves get these therapies to the patients that need them more quickly. So let me leave you with this. The last major breakthrough that happened in the treatment of cancer was chemotherapy, nearly 70 years ago. Immunotherapies are the new cutting-edge technology, and they have led to the advancement of treatment in multiple cancers. Unfortunately, they haven't made their way into the treatment of bladder cancer. Someone is going to help a lot of patients and their families. With your help, we can get our cutting-edge immunotherapy from the lab to the patients that deserve better than the unacceptable choices that they have today. We are Vivimune, and we are fighting cancer at the source.
Hi guys, congratulations, nice presentation. In your business plan, you laid out that you thought that this was going to go from being a fairly small $175 million market to a $4 billion market, which raised my eyebrows. That's a 23 times market increase. But in your presentation, you said it was going to be $11 billion, which is a 100x market size increase. Help me with the math of that and drill down into why you believe that.
Yeah, that's a great question. So the current market size is based on BCG, which is an old low efficacy off-patent technology, meaning that the costs of the therapy itself are low. But actually, bladder cancer has one of the highest costs per patient of any cancer due to the need to continually monitor for recurrence and give these new treatments. So that $4 billion figure that was quoted in the business plan was looking at a subset of patients in the US that we thought could be some of our early adopters. And the figure that you saw up here was global numbers of new patients every year. So that was a global figure. And since most of the acquirers that we would be targeting would be global companies, it made sense to look at that as well.
Okay.
Mm-hmm.
Okay. So my question has to do with risk, and I want to understand how you think about it. So I'm going to talk about a couple of areas, and the question is, so you could have scientific risk, regulatory. Let's say this just doesn't work out when you go into preclinical trials. Or it could be related to intellectual property, problems getting your patents. Or it could be competitive rivalry, others also working on immunotherapies at other universities and things. Or it could be the ability to raise funds, or it could be something else. So rank these risks from 1 to 4 in order to, giving me the greatest risk all the way to the lowest, and tell me why you think the one that's number 1 is the biggest risk.
Okay. So I think that certainly the scientific risk is there because, frankly, none of the rest of them matter if the science doesn't work. The funding risk, I would say, is less risky based on the fact that there are grants involved, and our chief scientific officer has been very successful with getting grants. So I put funding on the smaller side, even though, again, it's asymmetrical risk for investors, right? It works or it doesn't, but ultimately I would put the science piece further ahead than I would the funding.
Bob.
You hear me?
Yes, sir.
Great. All right, so I'm going to dig in on some of those risk categories. I'm going to start with the science risk, and I'm going to ask a couple of questions. First, as I understand it, one of the features of your drug is that you're directly implanting it in the tumor. How many tumors typically occur in a bladder cancer patient? Is it a single tumor or is it multiple tumors? And if it's multiple tumors, how do you know that you've delivered the drug to every tumor you need to deliver it to?
Yeah, that's a really good question. So normally in bladder tumors, it exists as a single lesion that you can see and you can inject.
And the physician identifies the tumor based on imaging and...
Yeah, they send a scope up there. Right now what they do is they send a scope in, they check out the tumor, they take a little biopsy, look at it and say, oh, that doesn't look good. And then they go back in and they scoop it out the rest of the way and then give a little chemotherapy. So they already have mechanisms to go in and access that tumor. Mm-hmm. Actually, in our talks with Dr. Neil Storr, he said it wouldn't be a problem.
All right, so let's talk about some of the other... You're talking about this as a platform.
Correct.
That has applicability to other solid tumor disease states. So let's talk about some of the other solid tumor disease states that you've identified, colon and breast. How often do those typically occur with a single tumor?
It depends on the stage of the tumor. Generally, breast tumors especially will appear as a single lesion that is relatively accessible for injection. The same with colon. You would see that as an initial tumor, and then you could go in with a scope once again and inject that.
Are you certain that in colon tumors there's typically only a single tumor? If it's multiple tumors and you only happen to address some of them, it isn't a very effective mechanism for dealing with the solid tumors. And second, I guess, is which is your second disease state? Is it pancreatic, colon, or breast?
Okay, those are good questions. With regard to the colon tumor, I'm not sure what the average number of initial lesions there are, but I can tell you this. In some of the studies we've done with bladder cancer, we've treated bladder cancer in the bladder in mice while giving a tumor somewhere else, and that tumor that was elsewhere also went away. Mm-hmm. Saying that an injection at one site can lead to dissemination of information all throughout the body. That's what the immune system's good at.
Okay, so I'm going to pass off in a second, but I have one more scientific question I want to ask.
All right, great.
Why are you doing large animal studies? Typically, in my experience, you can pretty quickly go from the small animal models that you've done to, trying to get into human clinicals. Why do an extra year on large animal studies?
We've been advised by some of the CROs we've talked with, as well as some of the members on our, advisory board, to pursue these large animal studies.
But why?
And one reason is that the component we have is a human molecule that doesn't work in mice. So.
Okay, and why didn't you use rat models instead of mice models?
It doesn't work in rats either.
Okay, great. I'll hand it off and come back.
So some of the benefits that you've called out are against your BCG is cost and efficacy. And so what you're claiming is you do 4 injections, and then it's going to treat the tumors from recurring for the lifespan, right? So do you have data to prove that? I see that you have, you did some tests 18 months after, you did an initial injection. So what data do you have to claim that it's going to treat it for the rest of the life?
Well, the lifespan of a mouse is about 2 years.
Okay.
So that's about as far out as we can go, is 18 months with a mouse.
But do you have enough data to prove that in humans, this is going to prevent reoccurrence, because a lot of your cost for BCG is every 3 to 6 months they have to administer that.
That's right.
So how do you claim that it's going to treat it for the rest of the life just based on?
Well, we claim it based on the best data that we have in the mice.
So how long is your clinical trial then? I mean, if you have to prove that it's going to treat, when you do human clinical trial, phase 1, how long is that study going to last before you can prove the efficacy?
So in phase 1, you don't actually have to prove efficacy. You can just prove safety.
Toxicity, okay.
Yes. Although we would probably do a phase 1b in which we wouldn't necessarily show that it improves survival or recurrence, because that would take long. It would take longer than we wanted to take. Instead, we would give the injection and prove that certain types of immune cells came in, per se. And I think that this preclinical data is exciting enough in itself to think that it has a lot of potential in humans.
All right. Very nice presentation. I'm curious about the IP, and are we talking about one provisional patent application that all this is based on, or are there others? And what other strategies have you considered around IP? Have you looked at freedom to operate and the risk of encroaching on other designs or IP in the space?
Yeah, so it's currently in the PCT phase of the application. We have considered splitting the patent up into multiple patents based on the advice of our IP attorney.
And have you looked at the ability to operate without infringing on anyone else's IP?
I haven't looked into that yet, but our IP attorney gave it a favorable review.
So just so you know, the fact that if you have a favorable review doesn't necessarily indicate freedom to operate. And let's dive in a little bit on your competition. ALT 803 and CV 301, we're both going after the bladder indications.
Yes.
And so are either of those immunotherapies? And if they are, how do they differ from your immunotherapy? That's question 1. Question 2, you mentioned that you have molecules that the mouse model doesn't, or a rat model doesn't work, and yet you're citing efficacy from the mouse model. I'm a little confused by that.
Yeah. Let me start with the first question, which was, wait, actually I just forgot.
Competitors, ALT, 803.
Okay, yes, competitors ALT, thank you. So ALT, it works in a different mechanism than ours does, in that it's not delivered locally, it doesn't stay there, and it doesn't elicit quite as strong as an immune response. It hasn't shown that preclinical data that we talked about before of being able to reject new tumors. Second, the CV
301.
Yes. Works by giving cells into the body that secrete particular antigens and then still giving BCG. So it's more of a vaccine in that instance. And once again, the data is not nearly as good. And the second question
Goes
was,
to mouse-
with
ma-
regard
efficacy-
to the difference
efficacy.
in mouse and humans, yeah. So the molecules are very much analogous. And we have the ability to make the human version of the molecule. And we have shown that it works on cells. We just haven't been able to put it in humans yet.
All right, but your advisors are telling you need to do large animal as a step to having some evidence of efficacy that's closer to human.
Yes, that as well as safety.
And yet you've sort of cited the efficacy in the mouse model as a strong indicator of the likely efficacy of the potential drug. And I'm confused if you have to do the large animal model to even be able to step to a human trial.
Yeah, that's a good question. So one of the problems with showing efficacy, say, in a large animal, is that there aren't tumor models for that. So you got to do the tumor models in the small animal to show that, oh man, this can destroy tumors, and then switch to the larger animal to get preliminary toxicity data before moving into humans.
Okay, let me ask. Okay,
So-
go ahead.
Just to follow on to that, I'm curious if that's a scientific decision or a demonstration of efficacy for the purpose of the acquisition. Can you address that?
Yeah, that's a great question. So that could provide an opportunity for another exit point, just as you're alluding to.
Right.
So,
I've got a little more of a macro level question, but it's riffing off these. So I want to understand the competitive landscape, not just with respect to bladder cancer, but let's add pancreatic and breast and colon. And I want to get a feel for the landscape. So are there hundreds of companies working on this, on immunotherapies, or dozens, or just one or two? Give me a feel for this.
Somewhere in between dozens and hundreds.
Dozens and hundreds.
So, and what's interesting about our technology is the method of action that Sean discussed, the local injection, versus a lot of what's out there are called checkpoint inhibitors. So they target a specific antigen on cancer versus what we have, which is it targets patient-specific antigens. So, and Sean's actually, he can tell you more about the work he's doing in the lab on that piece of it, but he's done some recent work that says our molecule will actually help improve the efficacy of some of the existing checkpoint inhibitors that are out there in the market.
But this again is confusing. You mentioned the polysaccharide as the mechanism to stick it to the tumor, and yet when I asked you about when you have multiple tumors in a particular disease state, that you had sort of a broadened efficacy. If the polysaccharide is the determinant that sticks it to the tumor, how do you get the sort of belief that you have sort of more general sort of applicability as well? They sound as though they're mutually contradictory. The specificity of nailing it to the tumor versus the general kind of efficacy throughout the body.
1 minute, judges.
okay, I think I have two questions there. So first question is, how does it stick to the tumor?
No, I got that.
Okay.
That seems to be the basis of your efficacy. I'm
Yes.
trying to understand why it has more general.
So the reason it works beyond that initial tumor site is kind of the beauty of the immune system. Just like if you get a flu shot in your arm, you're protected from the flu in your mouth. It's the same idea. So what we're doing is we're shining the spotlight by keeping it only where it matters. So we're not getting toxicity throughout the rest of the body. We're putting it only where it matters, drawing the immune system into the best source of antigens that the tumor has, or that our body can provide, and then letting them go out and find the rest of the tumor cells.
One last question. So, have you? So this is primarily focused on cure and preventing reoccurrence, right?
Correct.
Have you? I mean, if there is systemic memory of this, by injecting this, why not use it as a prevention?
So there
Use
has
it
to
like
be
a
there has to
vaccine.
be a tumor there first
You've got to have a tumor.
for it to know what to look for. That's the reason you can't use it like a traditional vaccine. It's a therapeutic vaccine, but your body needs to know what it's looking for first, which is what our therapy helps to do.